Hello, this is Scott Friedman in New York, here to discuss this month's Journal of Hepatology focus piece, which has the provocative title of "There are decades where nothing happens, and then there are weeks where decades happen." This quote from Lenin seems ill-placed, and it is a bit ironic that I'm using it to describe what will likely be the most profitable drug sector in pharmaceutical history. But it does underscore the incredible revolution that we're all witnessing in the development of interferon-free treatments for hepatitis C. And it gives me an opportunity to talk about two specific papers that really touch on critical issues around this new interferon-free world. The first is by Zobair Younasi and colleagues that looks at the potential or projected cost savings that come with interferon-free regimens, taking into account the cost of care, the cost of diagnosis, and of course the savings that will come from high rates of HCV cure. And this very nice modeling paper indicates that the cost for quality of life years adjusted will be about $15,000, which is far less than the $50,000 threshold that is often used to justify new treatments. The paper is very well constructed. It is, of course, a model, and this will need to be validated in clinical practice as these new interferon-free regimens are used. And, of course, a lot can happen, as we learned with boceprevir and telaprevir, where the side effects were considerably higher than what had been anticipated based on clinical trials. But still, it lends very encouraging information to the justification for continued development of interferon-free therapies for hepatitis C, with the hope and expectation that this will, in the long run, cut healthcare costs. And that's really quite important. And I think it's those kinds of calculations that in part underlie recommendations from the United States Centers for Disease Control and the Preventive Health Task Force, both of which recommended baby boomer screening for individuals between the ages of, or born between 1945 and 1965, specifically to detect asymptomatic hepatitis C in this relatively higher risk age cohort. Because now we have great treatments. They will be eradicating the disease in a large majority of patients, well over 90%, and they will do so at minimal adverse events compared to interferon-free regimens. So there's every reason to embrace recommendations for baby boomer screening of this type to detect hepatitis C, and presumably, therefore, to treat diseases before they yield complications that are both costly and highly morbid for patients and also for their families. But the path is not completely perfect. There is a study by Anna Locke and her colleagues that underscore the fact that in patients who are already hepatitis C treatment failures from conventional therapies, that they may actually carry over resistance to some of the direct-acting antivirals. And specifically, patients who have been null responders or failed therapy with interferon are probably non-CC IL-28 genotype, which of course confers relative resistance to interferon-based therapies. And so the bottom line for this study, without going into much detail, is that it tells us that for patients who either are prior non-responders or who fail to respond to interferon-free regimens, particularly using cimeprevir, there may be resistant testing required to ensure that the drugs being used are not going to confront a resistant hepatitis C virus strain. So treatment of hepatitis C is getting markedly simpler, and of course the cure rates are going to be much, much higher, but there will be nuances to its treatment that will require clinicians to really stay on top of issues around drug resistance, and of course the possibility of more adverse events in post-marketing use of the drug than we're seeing in clinical trials. Still, it's a remarkable revolution, and it's one that we should certainly embrace and not forget. It's also an interesting opportunity to think about which were the major advances that really brought us to the cusp of this new revolution. And in my focus piece, I emphasize at least three major discoveries that really, in aggregate, were the ones that I think established this new era of interferon-free regimens. The first discovery, of course, was the identification of the hepatitis C virus agent by Michael Houghton and colleagues at Chiron many years ago. And this was a remarkable event because it not only discovered a virus that had enormous public health implications, but they used a remarkably clever and revolutionary new method to discover the nature of the infectious agent without actually understanding its structure or isolating its nucleic acid directly. The second advance was the development of cell culture systems in which either replicons or full replicating viruses could be developed and established so that this would allow for high throughput drug screening. And the third discovery was the discovery of the structure of these HCV viral proteins that allowed for rational drug design to develop protease and polymerase and non-nuc and other inhibitors that could directly attack regions of the virus and cripple it without impacting on host tissues or host molecules. One of the other implications of these three discoveries is they really occurred concurrently and I would say synergistically between the pharmaceutical industry and the academic community. So for example, the discovery of the viral agent was performed in a pharmaceutical company, but it relied entirely on the characterization of cohorts by Harvey Alter and others at the NIH in order to have well-identified patients who likely harbored non-A, non-B hepatitis at that time. And without that, there was no way that the virus could have been isolated. And then, of course, the development of cell-based screening systems was also done in academic labs by Bartenschlager and then subsequent models by Charlie Rice, Frank Cesari, and other groups that really developed the cell-based platform to enable screening. And then, of course, the structure was done again in industry. So this really represents a really nice synergy between what was done in the academic sector and what was done in the pharmaceutical sector. And I'd like to think that this is one of the great examples where a healthy partnership between commercial entities like drug companies and the academic sector have ultimately yielded benefit to patients in a way that really can advance a field. So here we are at this really critical and exciting juncture of a new revolution in hepatitis C therapies. I hearken back to the quote by Lenin and emphasize that this is an era that probably will not pass anytime soon for those of us in hepatology. So we should enjoy the view and, of course, embrace these new approaches in hopes of really curing hepatitis C in a majority of our patients and sparing millions of patients the prospect of end-stage liver disease, the need for transplantation, or the risk of hepatocellular carcinoma.